Good afternoon, dear students. Now we are going to continue with the ion exchange chromatography. Till now we have discussed what is the basic principle of ion exchange chromatography, what are the basic types of ion exchange resins, okay, what are the different cation exchangers and ion exchangers and all that we have discussed. Now see how many types of ion exchangers are there. Cation exchangers and anion exchangers. Cation exchangers have positively charged counter ions which are electrostatically bound to the negatively charged functional group. Anion exchangers have negatively charged counter ions which are electrostatically bound to the positive functional group, like that. Okay, now if you want to separate, depending upon what molecule you are separating, you will use either polystyrene or whether you will use cellulose based resins. Cellulose based resins are used for separating large molecule, generally Cephαρose or Cephadex, or CMC, carboxymethyl cellulose. Now polystyrene is used for separating small molecular weight compounds, right? Now, but before we start with the procedure, we need to understand about the exchange medium, right? See here, the column is filled with the stationary phase. Okay, the stationary phase material is a resin or gel matrix consisting of agarose or cellulose beads which are covalently bound charged functional group, right? The stationary phase material may be polystyrene or it may be Cephαρose or Cephadex. Okay, depending up if you are using a cellulose based one, Cephαρose or Cephadex. Okay, is to be filled into the column. But most important thing is preparation of this exchange material. Preparation of exchange material is an important step. Why do you want to prepare it? Okay, so that it can be used. The first thing is swelling the medium. So whether you are using polystyrene or whatever as the exchange medium, it has to be swollen first. Why swelling is important? Swelling, the step of swelling the medium is called as pre-cycling. Why? Because the dry medium, the dry exchanger have the charged functional group, okay, buried inside. They are, and okay, so the charged functional groups are buried inside. The counter ions are not exposed. Counter ions cannot be exposed for exchange. Okay, so the functional groups, charge groups should be exposed. Okay, because we want that the functional group should be exposed on the surface of the polymer or the exchange medium, we have to swell it. Okay, now swelling of anion exchanger is usually done by treating it first with acid 0.5 normal HCl and then with a base 0.5 normal NaOH. Reverse with cation exchanger. Okay, so you have to first swell the anion exchanger or cation exchanger. Okay, and if they are if any metallic impurities are present, they are chelated by adding EDTA. Then very fine exchange particles are present. These fines actually they give you unsatisfactory resolution and decrease the flow rate. So the fine exchanges, finely powdered exchanges which we do not want, are generally separated. How? By putting, okay, by suspending the exchanger in large volume of water. And when the large polymers have settled down, the slow sedimenting fine material can be decanted. Okay, now you have to, so first you have to swell it, remove the you can say metallic impurities by chelating with EDTA, remove the fines by decanting them. Okay, and finally the exchanger has to be equilibrated with suitable counter ion. Okay, that is called as, okay, making your ion exchange medium ready. So before you make your ion exchange medium ready, you have to attach counter ions to the functional groups that are present on the surface of the exchange material. Okay, so what type of molecule you want to separate, whether you want to separate positive cation or anion, depending on that your counter ions will be present. Okay, so you have to treat the exchanger with different types of chemicals or reagents depending upon the desired counter ion. Say for example, if you want sodium ion Na plus as counter ion, then your exchanger is washed with NaOH. Okay, so wash the exchanger with different reagents. With NaOH, if you want to introduce sodium ion, if you want to introduce H plus ion as counter ion, wash it with HCl. If you want to introduce NO3 or NO as nitrate or nitrite as counter ion, wash it with NaNO3. Okay, formic acid, if you want formate as counter ion. So depending upon what counter ion you want, your resin ion exchanger is to be washed with different chemicals. Now your ion exchanger resin is ready, your column is ready, the stationary phase material is ready. So how to make it ready? First, swelling the ion exchange material. Then yeah, okay, so how to swell it? By using acid and alkali, or alkali and acid, depending upon whether it is a cation or an ion exchange resin. Then removing the metallic impurities by chelating with EDTA. Okay, removing the fines by decanting them, and finally treating the exchanger with chemicals like NaOH or HCl or NaNO3, depending upon what counter ions you want. So your ion exchange stationary phase is ready. Then the choice of the buffer. The buffer which you are going to use for eluting out your sample molecule depends upon whether it is an anion exchanger or cation exchanger. Okay, if okay, now see, anion exchange chromatography. When you are carrying out anion exchange chromatography, it is to be carried out with cationic buffers. Okay, and cation exchange chromatography is carried out with anionic buffers. Like that you have to take care of, right? Now so once your you can say stationary phase material is ready, the sample is introduced either manually or with a auto sampler into a sample loop of known volume. Okay, so the sample is introduced using an auto sampler or manually. The mobile phase, that is the buffered aqueous solution, carries the sample from the loop into the column that contains some stationary phase. The stationary phase material varies. It may be Cepharo, Cephadex, it may be polystyrene, whatever. Okay. Now having charged functional group attached with counter ions. The target analytes. Target analytes means the analyte, the molecule present in the sample, are retained on the stationary phase, but they can be eluted out by increasing the concentration of similarly charged species, as we have discussed earlier, or by adding the buffer having different pH. The analytes of interest which are separated out can be detected by some means like using a conductivity or UV visible absorbance or any other such property. And the chromatographic data system is usually needed to control all these analyte analysis. Okay. So loading the sample on the stationary phase material, allowing the mobile phase to percolate through the stationary phase. Okay, then your sample molecules get attached, get exchanged with the ion exchange resin, and then separating the target molecule by either increasing the same charged molecules or by adding a buffer having a different pH. And the analytes which are collected can be checked or detected typically by measuring their conductivity or UV visible absorbance, etc. Now usually we carry out gradient elution, okay, rather than isocratic elution. This point we have discussed earlier. Gradient elution could be stepwise or continuous, okay, but gradient elution gives you a better resolution, right? During anion exchange, the pH gradient decreases and the ionic strength increases. On the other hand, during cation exchange, the pH and ionic strength both are increased. Okay, so by altering the pH of the buffer, we can separate the molecule. Now regarding the, just a minute, this has been hanged. Just discuss the applications of ion exchange chromatography. Applications of ion exchange chromatography. It has several applications. Okay, number one, the most important use of or application of ion exchange chromatography is amino acid analysis present in a protein. Okay, amino acid analysis is generally done by using amino acid autoanalyzer, which makes use of ion exchange chromatography to determine the amino acids present in a protein. So amino acid autoanalyzer is used to analyze amino acids present in a protein. Ion exchange chromatography is extensively used to determine the base composition of nucleic acid. Base composition means adenine, guanine, cytosine, thymine, what are present. To elucidate Chargaff's rule, okay, ion exchange chromatography is used. For what? Determining the base composition of nucleic acid. It is used for complete deionization of water. If you completely deionize the water, remove all the ions, you can use ion exchange chromatography. Ion exchange chromatography is also used for softening of drinking water, to make the hard drinking water soft. So complete deionization of water, softening of drinking water can be done by ion exchange chromatography. Okay, and then there are certain biological applications or biological reactions where ultra pure metal ion free reagents are needed. Okay, so there are certain reactions or certain procedures where we require ultra pure metal free reagent, and this ultra pure metal free reagents can be commercially obtained by ion exchange chromatography. Okay. See, so in certain biological sample, trace metals are present. The trace metals are present below detection level. Below detection level of AAS means even atomic absorption spectrophotometer or atomic absorption spectrophotometry process cannot detect the metal because they are present below detection limit. These can be detected and removed by ion exchange chromatography. So apart from all above applications, ion exchange chromatography is used for separation or purification of vitamins, other biological amines, organic acids and bases. Okay. So that is all about the ion exchange chromatography.